Hello everyone. So based on my experience or being a student of genomics, I would like to tell you for setting a sequencing run, it is very important to plan the run. And for a successful sequencing run planning, there are several things which one has to take care of, but knowing the right amount of data which needs to be generated from a sequencing run is very important because based on the experience I can tell you sequencing is not about generating maximum data but generating the right amount of the data. And also most sequencing failure don't happen in the machine but they happen during the planning. So planning is very important, and for planning, we have to take care of several things, and one of the most important thing is how much sequencing data we how much sequencing data do we need. This we have to decide before starting the run. And why this is important? Because if you are knowing the right amount of data which we need to generate, then only you will be avoid wasting sequencing capacity. Because when you buy a very costly flow cell for a sequencing platform, you have a very specified data, a maximum amount of data that can be generated using the flow cell. So knowing the right amount of data generation needed for your sequencing experiment, then only you will be able to avoid wasting sequencing capacity of the flow cell. And also very importantly, then only you will be able to ensure sufficient coverage for your sequencing samples. And also your result will be reliable and accurate. And most importantly, you will be saving the cost and time. So for deciding the sequencing data before you start the sequencing run, you should know two key terminology. So one is genome size, that is total length of DNA in an organism. For example, if you are working with a human genome, so that means the size is 3.2 gigabases. For example, if you are working with bacterial genome, a typical bacterial genome is consist of roughly about 5 million bases. Similarly, if you are working with viral genome, for example, if dengue virus, somewhere around 11 kilobases genome size is. So genome size you have to keep in the mind. And the second term is coverage depth. So you have to decide your study required how much coverage depth, like 30x, 10x. So these two key terminology is very important in determining the sequencing data required from a sequencing run. So let's understand the coverage depth. So coverage depth by definition, it is average number of times each base in a genome is read during a sequencing. For example, if coverage depth is 10x, that means each base is sequenced or each base is read 10 times. If it is 30x coverage, then that means each base is read 30 times. So basically higher coverage will give you higher confidence. And in summary, coverage tells us how many times each base is sequenced. So now, since you know your genome size of the sample which you are going to sequence, and you have decided what is your coverage depth requirement. Based on you can easily calculate the right amount of data required. So very simple formula. Here it's go. The total data required is equal to genome size multiplied by coverage. So this formula we will understand based on some example. For example, if you are working with human genome, as I mentioned, the size roughly around 3.2 gigabases, and you have decided the coverage depth required is 30x. So in this case, the total data required would be 3.2 GB multiplied by 30. So 96 gigabases per human genome. So this information is very relevant. Based on that, you can easily calculate how much sample you can accommodate in a flow cell. For example, if your flow cell has X GB or X TB of the data, the maximum data that a flow cell can generate. So you can simply divide by this number and then you can get the number of sample you can accommodate in the flow cell. And now I would like to also mention the importance of 30x coverage. So you can see this is very standard coverage for human whole genome sequencing. This is considered as the gold standard for human whole genome sequencing. If you want to study any variant detection, clinical studies, if you're going to find some rare mutations associated with clinical phenotypes, then this coverage is considered as very standard. Let's consider another example. If you have, again, if you are working with human genome and you want only 10x coverage, in that case, you will require 32 GB of data per genome. So here you can accommodate more number of samples in the same flow cell. So here what is important is the 10x is cheaper, but you can say 30x is also safer. But it depends on the kind of study you are doing. So generally, this 10x coverage is being used for population studies, low pass sequencing, and also if you have some cost effective analysis to do or some kind of demonstration. So, but why 30x is preferred? Because it reduces sequencing error, it improves variant confidence, it handles uneven coverage, and as I mentioned, it is standard for human WGS. So I hope this small presentation will be will guide you in calculating the right amount of data. Thank you.